Good afternoon, all of you. I know it's in between lunch and I'm standing. I'll not take much time. We'll be quick. Just to update you, what are the new solutions which we are bringing up for your new advanced area of the oncology. So this is the first slide, which is basically says always we always strive to be the sample to insight solutions. Like you start from sample and you get the result. So in between there are multiple steps when it comes to molecular pathology. So always there are challenging areas when starting from sample, do nucleic acid extraction. So we always try to do whatever best we can offer here. So in this workflow, a recent thing which we have recently introduced, one platform. Currently we are having a lot of challenges when we do DNA extraction from FFP sample as well as from a liquid biopsy sample. So this is something which we have put in that. So it's a fully automated platform where it isolates DNA as well as RNA from FFP samples without any manual steps. It's involved, in manual step, there are a lot of manual steps are there when we work with FFP samples, and DNA is also very compromised with this. So it works with 1 to 24 samples. So either you can do one sample. It's a good thing is it's fully automated for cell-free DNA isolation, and you can go up to 8 ml of sample. Everything is fully automated with this. Then automated enrichment of CTC cells, and you can further isolate the mRNA from that. So it's an automated platform for, and we can isolate even DNA as well as RNA also from FFP sample. Same sample can be processed in that, and it can be connected to Cryosphere is a cloud-based system where it can be connected with this. So good thing is that it's magnetic bead based. So if you'll see there are multiple, we have different kits. I'm not going to all the kits, but you can see just to show it that we have 4 different kits. We have dedicated kit for FFP sample where we can, and in fact, in this we can do UNG treatment also to avoid the deamination of cytosins for those residues. And then we have cfDNA, and then a separate kit for CTC enrichment automated, so that your end result always goes good. Because when it comes to molecular pathology, always whatever will put the DNA, result depends on that. The better quality DNA, you'll get the better result out of it. And the thing is there, which is there, is a digital PCR. It's a advancement in the digital PCR chemistry. So in this, basically, it's a earlier we were having like droplet based. Now it's a nanoplate based. So good thing is the nanoplate based PCR is that it has already fixed partitions. Always the earlier challenge was that you are always having you need expertise to make the good number of droplets. But here is everything's partitions. You no need to put anything. You have to just load the sample. It will always, you will have equal number of partitions in the platform. So you'll have accuracy of the results. And then results also you can get in up to 2 hours. Starting from sample till results, you can get it. And in this, there are multiple formats depending on your throughput. You can have 1 plate, 4 plate, or even 8 plate system if you have the high throughput. And then we have in this multiple assays options also there. We have LNA based assays which increase the sensitivity and specificity. And then we have duplex assay also, so that in a wild type and mutant you can detect together in the same reaction chamber. And it's well tested. We have multiple solutions in this wet lab. We have almost more than 500 assays which are available right now, and some more in pipeline. So this is basically workflow. In this workflow, why we say once we work with liquid biopsy or FFP sample, you can do everything is automated. You can do extraction using EZ2 platform and then go to on the digital PCR. Everything is very validated and automated platforms are there. These are the newer things which has come in this. Now it comes to in this, what's happening basically, if you do EZ2 connect liquid biopsy, up to 8 ml also will do. There's no hands-on nothing. The many things are there where you increase the volume, you have to many steps are manual, but it's not there. Second thing, in digital PCR, you can see in this we can put reaction volume up to 40 microliter. The benefit of increasing reaction volume is that if you're low, you are having very low target, then also you can see in my reaction chamber more than 1 copies are there. But once in other platforms your reaction volume is low, suppose 20. So actual DNA are putting only 5 microliter, 5 to 10 microliter. So in that your copies are low, then you're limiting the amount of elevate. But here you can increase the elevate because we can go up to 40 microreaction. So increase your efficiency as well as sensitivity of the platform, I say. So in this, we have multiple options. So we have the plate also, we have 3-4 different plates are there. So starting from 16, we have 8,500 partitions, then 26,000 partitions, and then we have different formats also. If you have low volume samples, then we can change it on those. And then different reagents are there, not going through those. Basically, if you have multiple plates to go with different kind of sensitivity. Suppose you don't want only with blood and you don't want with CT like plasma, so you can go to 8,500 partitions also. So you will save cost also in that. So we are giving flexibility basically to the users. And apart from this, now it comes to we do have, apart from this personalized healthcare. So we do have like for brain, melanoma, colorectal, and for lung cancer, we have multiple solution. And this in this recently we have launched a new solution, which is Theraskin. It is well known. You must be knowing Theraskin is a worldwide. We are like in companion diagnostic. Is we are having multiple collaboration with the pharma companies. So in this recently the launch is this. In this we have launched a new kit with 42 mutations. In this, and good part is that the same solution works with plasma as well as FFP. And this in this we are LOD for T790 we target up to 1.8%, which is there in the currently. That's a good part. And fourth, last thing, the workflow, just to make the automated results are you can analyze automatically. Just a one click, you will get the results in there. So your competency is upon the level, so it reduces to that level also. So there's a good part in this. And the good part of this, as I told you, like first slide, so we have a lot of these kits are approved, FDA approved kits, and these are having companion diagnostics. Approval is there from FDA. So we have multiple drug approvals are there. And then it works with complete workflow, automated workflow is there. And for we are going up to 1% of sensitivity in case of lung cancer, EGFR. So both the kits and you can do automated result analysis with this. Not going very much in this. And then apart from this, we have the leukemia, we have the multiple solutions. So also been in myeloid with the MPN, AML, ALN, CLL. We have solutions in most of the areas where we are, and all the assays are basically based on the NCCN guidelines, which we assay has been designed, because these are being sold in global all the regions. Now we have another thing which is very good is NGS workflow will always work in that. So I'm inviting my colleague Amit here. He will just go through what are the NGS solutions which we have very quick as well as automated solutions in this. Amit?
Thank you, Naresh. So this is basically we are having the NGS portfolio wherein if you see here in this particular slide, we have sample to insight solution like from the QIAGEN, from sample collection to nucleic acid isolation. Then we have this library preparation solution, which can be sequenced on Illumina, Thermo, and MGI platforms. Then the sequencing run has to be performed on that instrument. And after this data, which we are having as a FASTQ file, can be analyzed using QIAGEN secondary analysis softwares, followed by the interpretation software like Ingenuity variant analysis, QIAGEN Clinical Insight Interpret, and other tools also we are having. Okay, so how QIAGEN is different compared to any others is that, first of all, let's see that what are the challenges which we are having in our targeted panels. We are having DNA, RNA, which we convert into cDNA, then we do PCR amplification. So during this PCR amplification, as you can see in the red color, that primer design constants are there, which affects the uniformity and specificity. And then also the PCR duplicates and errors are coming along with the biasness in the GC-rich region coverage. So these are being incorporated. We have incorporated the single primer extension SPE technology. That means we require only single primer for target enrichment. And we have incorporated the unique molecular indexes. And we have a proprietary chemistry to cover the GC-rich regions more efficiently. So just coming with the SPE technology, single primer extension in oldest fashion, like we have forward and reverse primer to amplify a target regions. This only reads between the targeted sequence and it is truncated at one site, right? So how it is going to differ is that in our scenario, this single primer extension, we are having we are only dependent upon a single primer that is working as a reverse primer, but we are having the forward primer as a universal primer here. So that is why we are able to cover the second part, which can be novel. So by using this method, we are not going to find the known mutations or known fusions, but we are also going to find out the novel gene fusions which are attached with a known gene or known gene partner. Then the second benefit of single primer extension is that, first of all, going to the dual primer approach for enrichment, we have to have about more than one tubes for the panels. But what happens? Hello. So what happens that we are having some exclusion regions where we are not able to design both primers, like either forward or reverse primer cannot be designed. So we are actually missing those regions. So coming with this particular single primer extension, we are having a single primer. So since we are not dependent upon other primer, we are having the complete region coverage. So for any panel, we are able to design more than 99% of the coverage here. And also we are having only single tube as a panel. So that is a benefit for you, that you are covering and not missing any particular mutation which could be of disease involvement, right? Here, next is that it is molecular barcode incorporation or unique molecular indexes, that we are actually attaching the molecular barcode as a adapter sequence here before the PCR amplification. So what happens is that after the PCR amplification, we are able to find out those amplicon sequences at the same position, that whether it is coming from the single PCR or from the unique amplicons. So if it is different one, then actually these are unique. If they are same, that means these are the PCR duplicates. So that is how we are going to identify the PCR duplicates, and this is going to help us getting a accurate quantification. So if you are calling a CNV, copy number variations in a target region, you are having more confidence on the data. The second benefit of the molecular tag or unique molecular index is that if you are having a particular mutation residing here, and if all of these amplicons are coming from the same amplicon after the amplification process, then this is a false positive, because other amplicon copies of the same amplicon is not having that particular mutation. But at the same time, if all of them are having, or more than 50% of them having that particular mutation, that then you are more confident that this was really a present in your original DNA or original amplicon. So that is why we are actually going to have more confidence on the SNVs or single nucleotide variations, right? And the third benefit of the GC coverage is that the unique proprietary chemistry which QIAGEN panel is having is that in this particular two different scenarios like CBPA and CCND1, they are having very high GC content regions, as you can see in this black bars, and the coverage is really very, very good, and everything is being covered. So this has already been validated, so that by using the QIAGEN panel, it gives you a better confidence, not only in the CNVs, not only in the single nucleotide variants, but also it is giving you the better coverage and uniformity of the data. So basically, you can use this particular panel on any input DNA, like whether it is very high, good quality input sample from blood, or it is FFP or liquid biopsy or cfDNA. In every panel, it is going to work the same panel, and you can use this panel on Thermo platform and Illumina platform and MGI platforms as well. So thank you so much for hearing us, and any questions?